Welcome to the first Center for Biosecurity Studies webinar for 2023. On behalf of the principal of the UWI Cave Hill campus, the CBS Advisory Committee, and the CBS team, we specially welcome our group of expertise presenters, those of you who are joining us for the first time, those of you who have been participating in our various outreach efforts over the years, whether you're joining us from Barbados, regionally or internationally, welcome. For the benefit of those who are joining with us for the first time, the Center for Biosecurity Studies, which I would refer to as CBS, is the sole entity at the UWI and within the entire Caribbean explicitly devoted to biosecurity. At CBS, biosecurity is defined as the science and practice of safeguarding lives and livelihoods, which yield a unique perspective on its importance to national and regional sustainable development efforts. The mission of CBS is to support these efforts to assess, to prevent, to mitigate where possible, and to respond to any threat that could potentially destabilize Caribbean societies. The Caribbean is comprised of several small island development states, and as a result, is vulnerable to a unique set of biosecurity threats, including climate change and its impacts like severe weather storms, tsunamis, rising sea levels, earthquakes, floodings. Then there are also food security, human trafficking, cybercrime, money laundering, inadequate waste management, poor sanitation, water scarcity, dependency on tourism, to name some of the major risks. CBS aims to represent a unique and comprehensive resource base and academic repository of expertise against these major inherent biosecurity threats. How exactly do we expect to achieve these? Through the facilitation of cross-disciplinary approach and cuts through the processes and thinkings at various levels, including international, national, and regional, in order to develop more effective and comprehensive solutions to the inherent threats to the Caribbean development and societies. I am Janice Brown, the Clerical Officer of CBS. I invite Professor Troy Lorde, Dean of the Faculty of Social Sciences and the Deputy Chairperson of the CBS Advisory Committee, to introduce you to this exciting webinar today. Bird flu, where are we now? Professor Lorde.
Thank you, Janice. Good afternoon, colleagues, and all of us following this webinar online, wherever you are across the world. Today, I have the distinct pleasure of offering these welcome remarks at the first Center for Biosecurity Studies webinar for 2023. I bring you warm greetings on behalf of the Faculty of Social Sciences at Cave Hill, and I want to thank each one of you for your attendance at today's webinar. Now, as part of my remarks, and you heard Janice actually give, in some ways, a preview of my remarks, some of my remarks, I want to say a little bit about the center and the center's work. Now, the Center for Biosecurity Studies at Cave Hill is really the first organization in the Caribbean to tackle multidimensional biosecurity issues and thereby attempt to stem the growing vulnerability of countries in the region to various biological threats, either occurring naturally or perhaps through malice. It is the first of its kind within the Caribbean, and it leads through several pillars: advocacy and outreach, education and training, research and consultancy, policy, entrepreneurship, and program development. The center currently offers professional short courses, and in the near future intends to roll out an MSc, an MPhil, and PhD in biosecurity studies. Now, over the last academic year, the center continued to focus its outreach and advocacy. It gave a total of 3 webinars, 1 public lecture, and 2 official addresses during the last academic year. In addition, The center was very highly in demand. It was invited to provide insight and commentary on many occasions, at least 12, on national TV, local radio, examining key global and regional biosecurity issues like COVID-19, the monkeypox virus, bird flu, air pollution, climate change, and its impact on infectious disease dynamics. So it also was invited to a number of regional broadcasts on UWI TV to mark World Wildlife Day and World Water Day. Some of the work of the center includes successful launch of 2 research initiatives, the 4th Invisible Border Initiative and the Big Ocean States Initiative. And this was to add to those that had already implemented the Caribbean Wildlife Initiative, the Caribbean Air Pollution Initiative, and the Waste Not, Want Not Initiative during the previous year. The first formative CBS research was conducted on climate and wildfires in Barbados, which was unprecedented. In addition, the framework and foundation were laid to direct future research and to build sustainable and suitable collaborations regionally and internationally. Now, the center, some other work of the center, as I mentioned, another pillar, which was entrepreneurship. It was the lead for the faculty in a submission to the inaugural Innovate KFL competition last year. And the submission Biosecure Inc. It will manufacture high quality green ecological bite and insect vector control products for residential and commercial uses. Prototyping of this product has garnered funding from Export Barbados, which is intended to cover the entire cost of prototyping. So workers are in train to bring this product to the market. Now, in today's webinar titled Bird Flu, Where Are We Now? We are going to hear from 4 global experts, Professor David Stalknecht and Doctors David Swain, Richard Webby, and Athis Kukan. I hope I didn't butcher your name. Apologies. Now, according to various reports, the world is going through its worst ever outbreak of bird flu. The highly infectious H5N1 strain is responsible for the deaths of hundreds of thousands of wild birds and millions of domestic ones. The World Organization for Animal Health has recorded almost 42 million individual cases in domestic and wild birds since the outbreak began in October 2021. So in some ways, this has been, I would say, a slow moving outbreak. We are increasingly hearing reports, but in some ways, you know, I didn't actually suspect that it was basically almost 2 years since this outbreak started. And I imagine for a lot of us listening to this webinar today, that would be news to you too. Now, almost 15 million domestic birds, including poultry, have died from disease, and another 193 million more have been culled. And worryingly, this strain has also been found in mammals. The World Organization for Animal Health has counted 119 outbreaks, although they have stated this is no doubt an underestimate. The World Health Organization has reported 870 humans have been infected with this flu over the past 20 years, and 457 have died. And their efforts are ongoing, really, to see whether it's mutated into a form that can spread amongst humans. Now, given our recent and ongoing experiences with the COVID-19 pandemic, it is certainly incumbent upon those doing research in the area to inform the public, a wary public, and I mean W-E-A-R-Y wary and W-A-R-Y wary public, about what we should expect from this outbreak, how it will be tackled by the experts and policymakers, what are the social and economic implications, and so forth. I look forward to hearing from our expert panel on these matters. Thank you, and I hope everyone enjoys the rest of this webinar's proceedings. Thank you. Back.
Thank you so much, Professor Lorde. At this point in time, I would like to introduce our first speaker and presenter for the afternoon, Professor David Stalknecht. Now, Professor David Stalknecht, he is a professor, in the Department of Infectious Diseases and the Department of Population Health at the University of Georgia. He's also the person behind the Southeast Cooperative Wildlife Disease Study, commonly known as SQUIDIS. On a personal note, Professor Stalknecht was actually my prospective PhD supervisor. And so we have maintained contact since many years ago, back in 2007. Now, his research interests lie in understanding the impacts of diseases on wildlife populations and the potential for these species to act as reservoirs or sentinels for livestock, poultry, and human diseases. Most of his work is oriented towards the epidemiology of viral and vector-borne diseases, including the epidemiology of blue tongue and epizootic hemorrhagic diseases in white-tailed deer, and also the natural history of avian influenza viruses in free-living duck and shorebirds populations. And his background in terms of education, he had a BSc back in 1973 from the University of Georgia, also a MS, Masters of Science, and a PhD from Louisiana State University. He has co-authored and authored over 300 peer-reviewed publications with almost 16,500 citations. And with no further ado, I'd like to introduce Professor David Stalknecht.
Thanks, Kirk, for the kind introduction, and I apologize for being late. I don't evidently understand time zones, and hopefully I can share a screen here. We'll see what happens here. Okay, is that being shared? You can now see your screen. Good deal. Get that on there. There we go. Okay, can everybody see that and hear me okay?
Could you just go to display settings?
Okay, Tice gave me the thumbs up, so we're good to go. Okay, what I'm going to start with is actually really covering the wild bird story of this, and there are some real experts on here that can handle the public health and the poultry part of this, and I really just want to stick with what we're seeing in the wild bird populations in North America, and really looking at this on the basis of really two questions. Will this virus persist or become a recurring problem? I think after this year, we may have answered the first one as far as it's persisting, and the Europeans have been giving us hints that this is going to happen in North America for about 7 years. So I think we have a situation now where we have an endemic virus, at least it looks that way. And really what I kind of will look at is, will this represent a significant wildlife disease? Flu has never really been considered a wildlife disease, but we are in a new world right now. We're seeing unprecedented mortality worldwide in a diversity of wildlife species, and it is of concern. One of the things we really need to kind of think about, though, is really what is the problem, and what species are going to be really affected by this. A lot of what we're thinking here is starting with what we know about flu. And in North America, this is all low path information. But we've been looking at this since the 1980s. We have a really good idea of the epidemiology of at least low pathogenic influenza, host range, and basically seasonal variation, temporal variation, and sort of transmission through water. And we're using this as sort of the basis of how we're viewing high path, to see if it follows this pattern or if it is likely to sort of follow this pattern in North America. Because if it does, it actually will give us some indication of risk, relative risk, and also risk associated with the spread, Central America, South America, et cetera, and what those in the Caribbean are really used to right now. And what we see with low path is basically, and it just is this one figure right here, is really a clear seasonal difference in prevalence across North America. And this does sort of reflect risk, especially with low path to introductions into poultry. So will this become a significant wildlife disease? And I think anyone in the wildlife disease arena right now will come up with the same conclusion. There is a lot to learn. As far as population effects, avian mortality is really difficult to quantitate, and denominators are difficult to estimate. One of the things we sort of, as a wildlife biologist, I think most people think we have a clear idea of population levels of a lot of wildlife species, but in fact, we don't. And the denominator is often very, very difficult. Even in sampling, a lot of times, this number does not come out. We got X numbers of positives, but no indication of what was looked at. So this is something that definitely needs to be improved. What drives infection and mortality? Can they be predicted? And the important thing with this and any wildlife disease, an infection does not equal disease. We have birds that are infected with flu, have suffered no clinical effects, and then have some that suffer mortality, really high levels of mortality. And this relationship may change seasonally between species, month, years, and flyways. Just because you don't see a problem in the spring doesn't mean you will not see a problem with that same species in the fall. Are there potential species-specific problems? And I think this is an area that we really need to investigate in detail. Impacts will probably differ by species related to pathogenicity and behavior. If you kind of look at what we see with waterfowl, we see differences in what we observe in ducks and geese. Geese seem to suffer a lot more mortality than ducks. Raptors, high path, unprecedented mortality in both Europe and in North America. This is maybe one of the more disturbing groups of mortality events that are occurring. Shorebirds and waders, at least in North America, sort of trickling through. But as far as potential species problem, probably high on the list. And then seabirds. And seabirds are probably receiving maybe less attention, at least in North America, than other species. But worldwide, some of the most devastating outbreaks have been in seabirds. And this certainly warrants a lot of additional study. And how may this affect the population? And there's varying ways a disease can affect a population. It could be subtle, and impacts may be restricted or more pronounced in hatchier birds. There may be specific age classes that are affected by this, and we need to kind of sort this out. And then again, where and when will this be a problem? Wildlife, poultry, humans, etc. Okay, what drives infections and mortalities can it be predicted? And I just want to sort of give you a little 3 case studies that we've been kind of looking at in North America. And the first one involves black vultures. And in all honesty, black vultures have never been implicated in the epidemiology of low-path influenza. Neither have turkey vultures, which is the other vulture that's common in North America. But starting in, I guess, February, is about a month after it was first detected in North America, we had a die-off of scalp, thousands of scalp in Florida. And we immediately started getting positive black vultures in Florida. In Florida, we've had positive cases every month from February through December. I don't think we have any from February this year, and it seems to be dying down. But this is an interesting situation. Basically, we're started in Florida, and if you kind of look through this, the states are on the y-axis. You can actually see an epidemic front going through as it moves through these populations. This is sort of a situation where mortality could not be predicted based on where vultures fitted in with low-path influenza, but maybe it could. If you start thinking about a totally naive population that has then, through scavenging on mortality associated with the outbreak, really is a new host population, host species. That theory is based on really the idea that this was a naive population. So we did just some quick serology, and really the serology actually shed some real light on this. Basically in Florida, where it was impacted, 67% of the vultures, the live vultures that we sampled, not the dead ones, but the live ones, perfectly clinically healthy, had antibodies to B. ELISA, with 67% testing positive to H5. And this is testing positive to the astrakhan antigen that is characteristic of the 2344. So a good indication that these birds are surviving, which is great. We went to Kentucky, where no cases have been seen to date, and you can see that it was basically a naive population, 7% B. ELISA, and 3% H5. And that could have been a migratory bird. Okay, with snow geese, it's a little different picture. We're not having this large-scale roost mortality, but we're having regional outbreaks. And there's a regional outbreak in the Midwest in spring of 2022, and also the late fall of 2023, with thousands of birds, hundreds to thousands of birds dying across the landscape. And again, looking at sort of the serology here, and actually saying, okay, well, really, how many of these birds were probably affected with this virus? They kind of put this mortality into perspective. And it needs to be said that snow geese it's actually an overpopulated species. There's millions of these birds. So actually, even if you have 2,000 or 3,000 birds die, it's a small percentage. But we did this again by looking temporally at sort of antibodies pre and post outbreak. And again, you can see a really increase in the number of birds testing positive for B. ELISA, and also more telling, how many of them are testing positive for H5, which again implies that some of them are surviving, at least maybe even a large proportion of them are surviving, which is good news when you kind of start looking at the resilience of the population. With blue-winged teal and mallards, we're getting into ducks, limited mortality. I mean, the birds die, but it's not generally big die-offs. There's singleton birds that are showing up. And what's kind of interesting with the blue-winged teal and mallards is these birds have a lot of exposure to low-path influenza. They're already, have seen a lot of these viruses, including low-path H5s. And this may be part of the reason that we're really not seeing a lot of mortality. That and some innate resistance, just for the fact that they're ducks. So these combined things that prevent mortality are difficult to evaluate. But long story short, we are seeing a clear indication of increased prevalence of H5. And we're also getting hints of, and we're going to have to do a few more years of this at least, of that same seasonal trend, where we're seeing a peak in September and October, is what we normally see with low path. Interestingly enough with this, if we kind of look at the prevalence of infected birds that we're seeing, it does mirror what we've seen with low path, but the difference is it's about 2 times higher than all the combined infections with low path we have seen in the past, which to me indicates that this is really a highly, highly transmissible virus. Okay, are there potential species-specific problems? And for this, I really want to just contrast what we're seeing in eagles in North America and what we're seeing in black vultures, because they're really very different things. Okay, as far as transmission goes and introduction in these species, with the eagles, it appears that these are individual introductions to eagles scavenging, maybe waterfowl, maybe other dead birds. With black vultures, it initially got into this population and actually was sustained through bird-to-bird transmission within the population. And it's kind of a sick scenario, but the vultures on the roost were dying, falling to the ground, and all their buddies were eating them. And it was just going on and on and on, and it's sort of a self-sustained. Whether it will be self-sustaining, who knows? The mortality associated with black vultures is catastrophic, at least on a roost standpoint, where a large proportion of them die. Long-term, we don't know what's going to happen. With eagles, it's a little different situation. There's large-scale mortality, but it's scattered all over the landscape. And with eagles, some of the mortality that we're seeing is actually associated with the breeding season, with birds bringing infected material to their nestlings. And in Georgia was local, but we saw a really major effect on the breeding success of eagles in parts of the state. So very kind of different scenarios. With shorebirds, this is a species we've spent a lot of time looking at. Shorebirds are kind of important potentially important for long-term movement of these viruses. They're really long-distance migrants, and they cover that area in a really short period of time. But we have not seen in North America any large-scale die-offs of shorebirds, which we're really happy about. There's been some sanderlings on the East Coast. I just looked at the USDA data, and I think it was a black turnstone and a willet on the Pacific coast recently reported in California. But these are the types of populations that we know are normally infected with low path. The transmission mechanisms are there. The interaction with maybe ducks is there. And we need to keep monitoring these birds. And equally important with these, we're talking about populations that are threatened. So flu getting in a population like this could really be a bad deal. And this really kind of goes to water birds also. We have not seen the die-offs in water birds in North America like they've seen in Europe and Africa and in South America, but they've been substantial and they have received limited attention. And in fact, the water birds and the shorebirds are not even on our national surveillance system. And we're really picking this up through independent researchers and state agencies and that sort of thing. But this is certainly one to kind of keep your eyes on. So how could this affect population and wildlife conservation and management? As we always said, we could have catastrophic losses. With seabirds, we've seen this. With geese, we've seen this. Annual large-scale mortality events with geese, these may occur seasonally, and especially if it gets to a point where the adults have some immunity, but the hatch-year birds don't. And if that's the case and this virus persists, we're likely to see these mortality events every single year. Age class specific mortality, as in geese, and recruitment effects, like we see in eagles. And this also has been reported from seabirds, from sandwich terns in Europe, where it affected birds on the breeding grounds, also gulls. We can have annual cryptic mortality and undetected events. And I think this is an area that's difficult to evaluate. But long story short, our surveillance, as good as it is or in North America, is not a sensitive surveillance system. And we miss a lot. We are literally hitting the tip of the iceberg. And this applies to raptors, and it also applies to wild mammals, which we've seen, which they reported in Europe for several years, and we reported here too recently. And with wild mammals in North America, it even becomes a little more tricky, because the one thing I failed to mention in this, we've been treating this as one virus, but we probably have had 4 separate introductions of 2334 in North America, 2 through the Atlantic and 2 through the Pacific. And there are at least 8 different genotypes of 2344 in North America based on reassortment with this virus with our native North American low path virus. And I'm sure you'll hear more about this in the next couple of talks. Subclinical effects, ducks and everything else, we don't know. I mean, we have done hunter-kill birds. We look in the brain. You can isolate virus from the brain of a clinically normal bird. And what effects that may have on a duck relate to migration, delayed migration, whatever, we really don't have a handle on. And then the perceived and actual risk to domestic poultry and humans. Scary situations can lead to bad management decisions when we're dealing with wildlife species. And we need to keep things into perspective. So where and when will this be a problem? This is sort of how we're advising things to go in North America. We need to shift from the current surveillance mode. We've lived in a world up to this to saying it's here, it's there, it's everywhere. And we need to get beyond that and understand why. Have clear objectives and know where to shift those objectives quickly. And that is really important. You just don't want to do the same thing over and over and over again. Develop an effective and adaptable outbreak strategy supported by science. Design it to document, inform, possibly reduce risk, and answer questions in a coordinated fashion. And answering the questions is the really important thing, because there are lots of them. Develop a culture, especially this is related to impacts on wildlife. Develop a culture where biologists, know how they can contribute to understanding impacts and epidemiology of influenza. They represent our most valuable collaborators and know what information is needed better than we do. Actually, collaborating with wildlife biologists, ornithologists in the field is critical. Poultry biosecurity is important, even if local risk from wild birds is not documented. We have surveillance, but as I said before, it is really insensitive. And on a local scale, it's not going to tell you whether they're infected wild birds in your immediate area or not. You should not rely on that as a basis of your biosecurity. And consider the possibility of human infection, but right now, do not go overboard. It scares people, it scares hunters, it scares biologists when you show up in a Tyvek suit. And you need reasonable protection, but don't go crazy. And I'm going to end on this slide right here. This is something that Andy Ramey wrote a long time ago, looking at the sort of the ecozones of influenza, or whatever you want to call them, the North America versus the Southern South America. And I just want to point out that the Caribbean, Central America, South America, as we know now, I mean, it's right in these birds routinely, gulls, shorebirds, ducks routinely visit these areas. These birds are going to be infected with flu. There's no question in my mind. And really some of the questions that you should have there is when it gets into this area, South America, Caribbean, we're dealing with completely different wildlife interactions that probably need to be looked at and considered. And with that, Kirk, I'm going to end it, right? Thank you.
Thank you very much, Professor Stallknecht. Very insightful observation so far with the panzoa that has been emerging all across North America. And I know you have questions. You can just place them into the chat. We will have a whole Q&A session right after the final presenter. So now we switch over to Dr. David Swain. Now we'll give his introduction. Dr. David Swain attended the University of Arkansas, Fayetteville, and the University of Missouri, Columbia, where he did his CSc in agriculture and DEM, Doctor of Veterinary Medicine. And he dual enrolled in graduate school during his freshman year, earning an MSc in veterinary pathology by the end of summer 1984, studying avian pathology under Dr. Arthur Bickford and the late Dr. Darrell Trampel. He furthered his graduate studies in veterinary pathology at the University of Georgia on Marek's disease under Dr. Oscar Fletcher, finishing the PhD in 1987. He's also dual boarded by the American College of Veterinary Pathologists and the American College of Poultry Veterinarians, ACPV. From 1987 to 1994, he was an assistant and associate professor on the faculty of the Ohio State University, Departments of Veterinary Pathobiology and Poultry Science, where he served as an avian pathologist. And during his first year as professor, Dr. Swain began conducting collaborative research with Dr. Richard Slimons on avian influenza in wild waterfowl. And this has become his passion and a lifetime career. And lifetime career indeed, because he is now recently retired. He's the former laboratory director of the Southeast Poultry Research Laboratory in Athens, Georgia, or the SEPRL. And the SEPRL is the in-house USDA high biocontainment lab for research on poultry viral diseases, including avian influenza, and is an international reference laboratory for avian influenza and collaborating center for research on emerging avian diseases. He has also served on the World Organization for Animal Health, or the OIE, international committees to update the avian influenza chapters in Terrestrial Animal Health Code and Manual. He served a sabbatical with WOAH, formerly known as OIE, in Paris for 16 months to study highly pathogenic avian influenza control programs. He was also the chair of the executive committee of OFF LU, the joint OIE and FAO organization of the United Nations Animal Influenza Network. He's participated in missions and conferences on avian influenza control, biosafety, and biosecurity in over 44 countries during the past 15 years. He has published over 285 peer-reviewed papers, 111 book or preceding chapters, and has given over 284 invited lectures on poultry health issues. He has also served as the editor of the textbook Avian Influenza. He's the associate editor, editor-in-chief, and the editor of forthcoming Animal Influenza. He's also on the editorial board of Avian Diseases, a journal, advisory board of avian pathology, and is an associate editor for influenza and other respiratory diseases. He has also served as the past president of the ACPV from 1999 to 2000, and on the board of directors for the American Association of Avian Pathologists, AAAP, from 1998 to 2000. So without any further ado, I present to you Dr. David Swain.
Thank you, Dr. Douglas, for that introduction, and I appreciate the opportunity to talk to you about high pathogenicity avian influenza. And my comments will be directed to just the poultry aspect, and then we'll cover some clinical signalment pathology, control, and end with talking about vaccination in my short allotted time. So when we talk about avian influenza, the ability of these viruses to cause disease, we categorize them into two broad categories based upon disease production in chickens, but it's really similar in turkeys and other gallinaceous poultry. This first category is the low pathogenicity viruses. They generally produce a local infection replicating in the epithelial cells of the respiratory tract and the GI tract. But also they can have impact on breeders. So drops in egg production can also occur with these viruses. Generally, you can recover the viruses easily from the respiratory tract, especially the upper respiratory tract and the digestive tract, for example, the cloacal swabs. And you can also find these viruses in infected hens on the surface of the egg. An example would be the H9N2 low-path AI virus, although they can be any of the 16 hemoglobin subtypes. Here's just a picture of an outbreak of low-path AI in meat turkeys in the U.S. from about 20 years ago. You can just see at a low power level that you have some turkeys in this that are kind of hunched over. They're not feeling good. They're lethargic, and they actually have closed eyes. Some of the turkeys, when you look at them directly, will have filling of the infraorbital sinus with mucus and debris, fibrin, et cetera, because they usually have a secondary bacterial infection with the influenza infection of the nasal cavity. In laying hens, this is a picture from an experimental infected chicken. There will be involution of the ovary and the oviduct. So depending on the stage of infection, there would be a decreased deposition of calcium in the eggshell, so it'll be soft-shelled or cracked eggs. And then as the ovary involutes, you'll see the destruction of the blood vessels as they involute with hemorrhages and the ovary quits function. The other broad category are the high pathogenicity viruses. These are the viruses that produce a systemic infection. It's usually a very deadly disease, which is caused by some of the H5s and some of the H7 influenza viruses. Generally, you can find the virus in almost any tissue in the body of chickens or turkeys. An example would be the current outbreak of H5N1, high path AI. So what you would see in infections in the field or experimentally, and these are pictures of chickens I think I'm going to have, you'll see the birds are lethargic. In this case, it's been sitting out in the activity. You can see around the eye is swollen, so there may be edema around the eye. You may actually have hemorrhage from the eye. You may see some birds that will have hemorrhage of the comb and wattle. This is from ischemia. It's plugging of the blood vessel from activation of the clotting cascade. You will see also hemorrhages under the scales on the legs and feet. In internal organs, a very common lesion would be necrosis, which would have pale spots in the pancreas, or you might actually even have hemorrhage in the pancreas. And then multiple visceral organs in the skeletal muscle, you'll have these petechia to ecumenic hemorrhages because in general, these high path AI viruses have a propensity to replicate in endothelial cells throughout the cardiovascular system, causing necrosis of those lining endothelial cells and hemorrhage into tissues where they are replicating. Just a little bit of data. This is from a study done at Southeast Poultry by Mary Panthine Jackwood and Erica Spackman and other colleagues, just to say that the signalment may vary depending on the species and the exposure time. In this case, in chickens, generally the chickens became infected on inoculation, and the death time was very quick. Usually by 3 days, they're all dead. The first ones will basically after exposure will die in the first 2 days. Generally, either you have no lesions, you may just walk in and find them dead, no promontory clinical signs, or you may find them in severe lethargy or having ruffled feathers. The few birds that may survive the very peracute stage may have cyanotic or necrosis of the combs and wattles and hemorrhages on the legs, periorbital edema and lethargy, just as I had shown you in the earlier pictures. With this virus in turkeys, it was a little bit delayed from Erica Spackman's study. No apparent clinical signs until right close to the time of death, and they would show severe lethargy, huddling, ruffled feathers. The few turkeys that would survive up to even, I think, 6 or 7 days generally had trouble walking and had neurological signs. And generally, if you look at waterfowl, let's say domestic ducks or geese, neurological sign is one thing that's a little more prominent in them. They tend to survive a little bit longer than the chickens and turkeys. So if we look at control strategies in poultry, eradication is the historical strategy for high path AI in the world as defined by the World Organization for Animal Health and most countries' national veterinary authority. And generally that involves in an outbreak enhanced biosecurity to try to prevent the high path AI introduction on those naive farms, or if it is a positive farm, to keep the virus on that farm. And so movement control is essential. The other thing about the enhanced biosecurity is it really you have to look at the birds, and if this virus, it's the entire building. So the door is the site or the line of separation where you need to consider that anything outside that door of that barn is going to be contaminated versus inside is the free zone. We also have to have in that eradication strategy good diagnostics and surveillance so you can quickly find that high path AI, and then a culling process of stamping out to eliminate the infection on those farms, along with safe disposal of those carcasses and cleaning and disinfection. And all this involves an education process where all individuals who have contact with this process will know their responsibility and have a high compliance rate for that biosecurity process. So if we look historically back to high path AI, we used to call it fowl plague or fowl pest, grip. And just for the 1924-25 outbreak, which is the first one we had in the United States, Dr. E.L. Stubbs at University of Pennsylvania made a couple of statements which kind of sets the tone for how we globally have dealt with high path AI in poultry. He said it's capable of causing such destruction of the poultry population as to be of economic importance in diminishing the food supply. And this is back in the 20s when all poultry production were usually a secondary product from farms, and usually people had less than 200 birds on the farm. Although at that time there were some layer farms that were specialized. I think the largest layer farm at that time was actually in Petaluma, California, and they had 5,000 birds, which would be a very small flock in today's standards. The other thing Dr. Stubbs said was the dangerous character of this disease, warranted the radical methods for complete eradication within a few months, which sets the stage for using the stamping out program, which had been used for other livestock diseases. Officially, it was implemented for rinderpest, and using that modified program for the eradication of highly pathogenic avian influenza. So the control since the 1920s has really been the stamping out programs, and clearly stamping out infected flocks was the very core of that program, with individual countries deciding whether dangerous contacts, contiguous properties, or zones of 1 to 3 kilometers, if everything should be killed within that zone. But really, what has changed since the 1920s? Well, we've seen a geometric increase in poultry production with specialization, integration, and increase in the size of farms and decreasing the number of farms. We've seen that eradication programs over time have become more expensive, and they've also faced some political and consumer burnout from that financial support to do them. Just a couple of examples here. The U.S. eradication cost for the 24-25 fowl plague outbreak was $14.2 million, adjusted for inflation of 2017. If you look at the 2014-15 H5N2 outbreak, that was $850 million for eradication. And the current outbreak, the 22-23 high path H5N1, we're running somewhere around $1.7 billion, of which it is not eradicated currently. Also, there's the animal welfare issue of the destruction of non-affected poultry and elimination of high-quality protein sourced by the stamping process. And generally, the problem of the reactive nature of the stamping out program, because you can be too late in the stamping out process where the transmission has already occurred off the farm before the diagnosis and quarantine and depopulation. That's why the essence of the diagnosis and how quickly potential flocks are reported, tested, and determined in quarantine are critical in controlling this disease and reducing that onward transmission. So when we look at the broad essence, and we're going to talk about just the rest of the time about this one virus that we are facing today, it's the 2344B clade of a Eurasian virus. In really the past 3 to 5 years, it's really become an issue of the virus being endemic in some wild bird populations that migrate across Asia, Africa, Europe, North and South America. So that has really changed the paradigm for the introduction of virus in poultry and onward spread of that virus in poultry populations and the control programs. So the issue is, globally between two issues. One is the virus being carried by wild birds, so they being a continual source, and the virus also being spread by poultry transport. This has made the outbreaks become increasingly common in poultry across these continents and has even entered into South America. So the process of stamping out elimination really has been the preferred control method in all of the developed world, and it's the process by which WOHA says we should follow. It's really not sustainable with the increased outbreaks in poultry we're seeing today and the reoccurrence of these outbreaks across even in our developed countries. This has become a huge economic burden, just thinking of the $1.7 billion that the U.S. government has paid for, but also the losses that farmers and companies have had in their livelihood in dealing with this outbreak. And so it has also threatened food security. Just as an example, in the United States, table eggs, which normally sell for around a dozen or about $1, but right now they're upwards of $5 for a dozen, so the supply is quite shortened. So biosecurity is that frontline defense. So in simple terms, biosecurity is really informed common sense, keeping pathogens away from poultry and poultry away from pathogens. So really our goal is twofold. One is to prevent the introduction and spread of this invisible pathogen, the avian influenza virus. And it's done by components that in total involve structural barriers, for example, poultry raised inside barns, so it's the walls, the doors, et cetera, that separates them from the potential virus. And then it's also the procedures, and this is very critical, the procedures that are followed by the farmers in preventing the movement of the virus from the outside contaminated environment into the facility, which would begin the infection and spread within that farm. In the same way, if you have an infected farm, you also have to practice biosecurity to prevent that virus from moving off the farm into other farms. But the big question is why is modern poultry production, do we still see these outbreaks? And I think it's very, very simple. It's conceptually very simple, is that biosecurity reduces but does not eliminate risk. The better you can practice the biosecurity to the highest degree, the greater your reduction of risk. If you don't practice that biosecurity, you will increase the risk and can eventually, if the right exposure there, have infection of your poultry, either at the village level or the backyard or even in commercial poultry. So in general, the control strategies for high path AI are based on the subtype and the pathotype, that's this lower high path, and in the individual country, what are the specific requirements of the country. Generally, the consensus is elimination from poultry by stamping out is the preferred strategy. But with this particular lineage, this goose guangdong lineage, which has been around since 1996, when it first was identified in domestic geese, the outcome there varies on the individual country, what they've experienced with this virus. So initially, with the immediate introduction, most countries began an elimination process by stamping out, in which many countries were successful. But some countries were not successful at that immediate elimination because of a variety of features. For example, it could be they had limited veterinary services in the government or in the private industry. Maybe they had restricted financial resources or didn't have the diagnostic capacity or the speed of the diagnostics, or maybe they lacked the logistics to be able to do an eradication stamping out program. Therefore, in some of those countries, the virus became endemic in poultry, or it was eradicated and then reintroduced back into the country multiple times. In some of these countries where the virus became endemic, vaccination was undertaken. In fact, there's a total of 15 countries that have begun vaccination in the last 2 decades ago, 2005 to 2010. And they used either a preventative vaccination program, an emergency vaccination program, or a routine program as a control measure to maintain food security for the production of birds. Well, as this virus has progressed over time, although initially the ecology part in wild birds was not well developed and there was not the massive spread, today with the 2344B and the ecological changes that have already been mentioned by Dr. Stallknecht, is that this virus is endemic in some wild aquatic bird species, which makes spreading, and it is a seasonal thing. It may produce a continued threat for introduction into poultry. So what can vaccination do? Well, vaccination increases resistance to avian influenza virus infection, which experimentally what we can do is show that it takes a much higher dose of virus to produce an infection in poultry. So in a well-vaccinated flock that's properly done, the resistance may be sufficient that it will prevent infection within that flock, and that's the goal. However, in some flocks, if the vaccination is not optimal and not enough birds actually develop an immune response or the vaccine doesn't match the field virus, there may be some infections among birds, but even in those infections, there's a reduction in the ability of that virus to replicate, and lower amounts of virus are shed in the respiratory and GI tract. What this does is it prevents disease and death in the poultry, and then it also reduces environmental contamination by reducing replication. That reduces the spread between premises and reduces transmission between birds. This improves animal welfare overall. So the vaccination, where it has been used, is an additional layer of protection on top of other biosecurity measures, but the vaccination itself does not replace those biosecurity measures. It's an adjunct, it's an addition, it's a supplement. So in October this year at the World Organization for Animal Health, a conference was held sponsored by the International Alliance for Biological Standardization to look at, can vaccines and the vaccination process gain wider acceptance as a control tool and help in this control and hopefully elimination of high path AI in many more countries. And just looking at the terrestrial animal code, which influenza has a chapter, it clearly states that vaccination by itself, as long as we have appropriate surveillance and program in place, should not inhibit trade or the distribution and sale and consumption of poultry products. But with the continued outbreaks, and I guess this particular epizootic sort of began in the fall of 2020 and has spread to 5 continents, the extent of this and the evidence of these increased outbreaks and large number of poultry being infected and destroyed has really brought to the forefront a discussion about, should we look at additional tools like vaccination to help in control and elimination of this disease? So the vaccination should be viewed as an extra layer of protection. It reduces that quantity of circulating virus, as I mentioned earlier, and the number of farms that become affected, of which stamping out would be required, would be reduced also. And I think for individual countries that are just facing this dilemma is that stamping out programs are very expensive. They're complex and they're very labor intensive. And they also require a lot of planning. There has to be exercising of those. So I think the question each country, including those in the Caribbean islands, will have to ask themselves, can your country continue with just a stamping out program, or do you need to examine vaccine and vaccination as an additional tool for your country? So in conclusion, I'll just say that this new clade that has emerged and has caused, at least in the Caribbean, wild bird infections in Cuba are the first ones, but also infections in Central and South America, both wild birds and in poultry. It causes severe disease in chickens and turkeys with near 100% mortality in unvaccinated poultry. The clinical presentation usually is a sudden death or severe lethargy, hemorrhagic disease, or even neurological signs or lesions. Stamping out programs have been the primary focus in most countries, but some have added vaccination as a management tool. And there is a global discussion and reevaluation of vaccination as a complementary tool to use with stamping out programs. And this is going to be critical for each individual country in the Caribbean to examine their veterinary structure. Can they do stamping out programs, or do they need additional help by using poultry vaccination as a supplemental tool? But if you do use vaccination, you need to make sure that you match the vaccine antigenically close to the outbreak virus, so you'll have predictable protection, because poorly matched vaccines do not provide predictable protection. There's been a lot of lessons learned through many countries throughout the world in management of the H5 as well as H7 high path AI, by use of vaccines allowed maintenance of food security and rural livelihoods.
Thank you very much, Dr. Swain. Another informative presentation. And I think the link with vaccination and trade and removing that barrier is definitely something quite intriguing. And as the situation evolves regarding vaccination and how we can add layers of security to the biosecurity measures that are currently in place, it's definitely food for thought. So again, you can put your questions in the question and answer section, and we will get to them once we've finished with the presenters. So now I turn to the next presenter, Professor Tej Kuyken. He's a professor of comparative pathology at Erasmus Medical College in Rotterdam, the Netherlands. And his research focuses on infectious diseases at the interface between humans, livestock, and wildlife. He is a professor of comparative pathology at the Department of Viroscience of the Erasmus University Medical Center in Rotterdam, the Netherlands, where he performs research on emerging viral diseases such as avian influenza and MERS using a One Health approach. He is the coordinator of Zoonosis in the Night, a Dutch project to evaluate the potential of viruses in bats to spread to people and domestic animals. He's also the co-coordinator of Delta Flu, a European project to determine the key factors determining spread of avian influenza between poultry and wild birds. And he's also the past coordinator of Antigone, a European project investigating the underlying causes for the emergence of infectious diseases from animal reservoirs to humans. And he's co-authored and authored several publications with over 33,000 citations. He's also won several awards, the University of Saskatchewan Scholarship, also the Robert and Virginia Roche Visiting Professor at the University of Saskatchewan. He is a recipient of the Journal of Comparative Pathology Plenary Lecture, and he's also the recipient of the 2013 Mid-Career Excellence Award from the American College of Veterinary Science. So without any further ado, Professor Tej Kuyken.
So thanks very much, Kirk. If you can load the presentation, then I will. On the effect of this 2344B clade virus on mammals, and look very much at the last 2 years. So next slide, please. So in the last 2 or 3 years, this virus has been found in 20 species of mammals worldwide. and it's likely that tens of thousands of mammals have been affected by this virus. Most of them have been carnivores, as you can see in this table from a recent report of the European Food and Safety Authority. But not only carnivores, also artiodactyls, marsupials, and cetaceans have been affected by this virus. Next slide. And if we look just in the United States, there's a very nice map produced by the US Department of Agriculture showing the distribution of reported positive cases in mammals, showing that 12 different species of mammals have been affected, and there have been around 100 cases reported. But as Dave Stallknecht said earlier, no doubt there are many more mammals because they are cryptic species that have been affected. Next slide. The reason that carnivores are so often affected by this virus is because of their habits of eating wild birds. And currently, some of those wild birds are infected with the virus. In this way, these carnivores get quite a substantial dose of virus by feeding, and they can become ill. Here's a photo of a red fox that has taken a female widgeon, one of the species known to be infected by this virus. Next slide. Important in carnivores that become ill from this virus is respiratory signs. And these respiratory signs are linked often to a severe inflammation of the respiratory tract. And here you can see the lungs of a fox that was infected with this H5N1 virus, showing its dark red color and liver-like texture of one of the lungs, really severely inflamed. Next slide. But we mustn't think of this virus as just being a respiratory disease in mammals. It often can spread to other parts of the body, particularly to the brain. And many mammals infected with this virus present with neurologic signs. And therefore, in carnivores, rabies and canine distemper may affect them. And now we need to consider this HiPath AI in the differential diagnosis. Here you can see at the microscopic level the effects of this virus infection. In the left column, the lung and the brain stained with hematoxylin and eosin, showing the inflammatory changes as a result of the virus. And in the right column, again, the expression of influenza virus antigen in these same tissues, showing that this virus infection is causing these lesions. Next slide. So until now, I've spoken mostly about infected mammals, and those have been only individual mammals that have been infected, or at most small family groups. For example, sometimes a small group of foxes are infected because they're related to each other and living close together. However, recently there have been several mass mortality events involving this virus and mammals. And the first one I want to discuss is this unusual mortality event of harbor seals and other pinnipeds along the east coast of the United States in Maine. And this was first seen as an increased mortality of harbor seals in June and especially in July along the Maine coast, as you can see in the graph on the left. And here on the right is an example of such a stranded seal. Next. So out of 25 harbor seals and 4 gray seals that were examined for high-path AI in this period. 15 harbor seals and 2 gray seals tested positive, and they showed lesions that fitted with their clinical signs, again, respiratory and neurological. And one of the sequences from the seals had a mutation in the polymerase gene E627K, which is a mutation that is known to be associated with mammalian adaptation. With so many seals infected by this virus, one doubts whether they all could have been infected individually by contact with an infected bird. And the authors of this report concluded that transmission was likely occurring either through environmental transmission, so virus, for example, on the rocky shore where they were hauling out, or by direct contact between seals, that is, seal-to-seal transmission of the virus. Next slide. So another mass mortality event in marine mammals, but now in 2 different species, and in the Pacific Ocean, not in the Atlantic Ocean, was an outbreak in sea lions along the coast of Peru in the first 5 weeks of 2023, when over 600 South American sea lions and a much smaller number of South American fur seals were found dead, sometimes with 100 animals found floating dead together. A number of these sea lions were tested, 12 in total, and 9 of those were positive for this H5N1 virus of the 2344B clade. Again, the animals showed respiratory disease, and they presented with respiratory signs, and a number of them also with neurologic signs. And with regards to the route of infection of these sea lions, the authors concluded that we cannot exclude direct transmission among sea lions due to their colonial breeding and because many animals died simultaneously in groups. Next slide. So the final mass mortality event in mammals I'd like to discuss is this outbreak, not in free-living mammals, but in farmed American mink in Spain, in the northwest corner of Spain, in the province of Galicia. And there, in October, in a mink farm with 52,000 mink in 30 partially open barns, higher mortality was observed, and this mortality rate increased until it peaked at 4.3%, meaning that over 2,200 mink died in a week in the middle of October. These mink also presented with respiratory signs and neurologic signs, and they also showed on pathology severe pneumonia. And of the 15 mink tested, 14 were positive for this virus, and the mink viruses that were examined more closely showed 8 to 9 amino acid differences between with viruses obtained from the most closely related bird viruses, and this included a mutation in the polymerase B2, namely T271A, which is again known to increase the polymerase activity in mammalian cells. Next. So the question arose where this virus entered into the mink farms. The mink were fed partly on poultry, on raw poultry byproducts, but in the period that this event in the mink took place, there had been no high-path AI poultry outbreaks in that region. However, in that region, there were several cases of high-path AI in seabirds, namely in gulls and in gannets. And these mink were housed in partially open buildings, as you can see in the photo on the left, and in this way may have been in contact with wild birds. And in the figure below, you can see a cartoon of a mink shed showing that the way that mink are typically housed means that not only wild birds, but also wild mammals, as well as wild bats, have free access in and out of these mink sheds so that there can be exchange of virus between wildlife and farmed mink. Regarding how it was possible that so many mink became infected with this virus, the authors concluded that our findings indicate onward transmission to other minks based on the increasing number of infected animals and the progression of the infection from a small area that was initially affected to the entire mink holding. Next slide. So this brings me to the question of how efficient transmission of an avian influenza virus between mammals can take place. And this question is often answered for humans with regards of the concern about an influenza pandemic. And one factor which is important is where the virus prefers to replicate in the respiratory tract. And one can conclude that efficient transmission of human influenza viruses, so the influenza viruses that are normally present in the human population, is linked to a preference of the virus for the upper respiratory tract and the trachea, as shown in the left cartoon, whereas inefficient transmission of avian influenza viruses is linked to a preference for the lung. Next slide. So this question can be studied in different ways. One way can be to examine the respiratory tract for receptors for influenza virus. And influenza viruses of humans have a preference for receptors in which the sialic acid is linked to the underlying saccharide by an alpha-2,6 link, whereas influenza viruses in birds in general have a preference for receptors in which the sialic acid is linked by an alpha-2,3 link to the underlying saccharides. So that's one way of looking at this. In our laboratory, we've chosen to look at it in a different way, namely by studying where the actual influenza virus attached to mucosa in different parts of the human respiratory tract. And this is shown in these pictures here. The attachment is shown as a red-brown staining of the influenza virus to the surface of the mucosa. And here is the outcome of a study of two human influenza viruses, H1N1 and H3N2, in the top two rows, and an avian influenza virus, H5N1, in the bottom row, for different parts of the human respiratory tract, going from the upper respiratory tract and trachea on the left, right down to the cell types in the lung, the alveoli and the macrophages on the right. And the important conclusion from this study is that human influenza viruses prefer to attach to the upper respiratory tract and trachea, as you can see by the red-brown staining in the on the left of the two upper rows. In contrast, H5N1 virus in the bottom row does not attach at all to the trachea of humans, and it only attaches to mucosa deep down in the respiratory tract, mainly in the lung. Next slide. So because we study avian influenza virus pathogenesis in laboratory animals to understand the disease in humans better, it's also useful to know what the pattern of influenza virus attachment is to the respiratory tract of the laboratory animal species that we use. And you can see in this overview that of the species that we often use in the laboratory to study influenza in humans, the mouse, the ferret, the macaque, and the cat. The ferret, macaque, and cat have a pattern of virus attachment which is more or less similar to that in the human on the top. But in contrast, the mouse has a very different pattern of virus attachment, where avian influenza virus actually attaches quite well to the mouse trachea. So from that point of view, the mouse is not a good model for influenza in humans. For many other parts of the pathogenesis, it's a very good model. Important here is that the similarity of the pattern of virus attachment in the ferret, which you can see the middle species, the similarity between that and the pattern of virus attachment in humans corresponds to transmission experiments that have been carried out in ferrets, where avian influenza viruses transmit not at all or poorly by air between ferrets, whereas human influenza viruses transmit very well by air between ferrets, which is the same as in humans. So in that sense, the ferret seems to be a very appropriate model for studying transmission of avian influenza virus in humans. Another additional study that we've done that I don't show here is that we've also studied the pattern of virus attachment in one of the species that was recently affected, the harbor seal. And interestingly, the harbor seal has a different pattern of virus attachment of avian influenza virus than most mammals. Namely, it's similar to the mouse, where avian influenza virus actually attaches very well to the trachea of harbor seals, which could explain why in the past there have been several outbreaks of avian influenza virus in harbor seals, causing quite high mortality in that particular species. Next slide. So if I just simplify the comparison of avian influenza virus attachment to the respiratory tracts of different species, I show it in this graph here, and this is also related to the outbreaks that I've just mentioned. So you can see here that high-path AI H5N1 attaches poorly to the human trachea, but well to the human lung. Similarly, in the ferrets, poorly in the trachea, well to the human lung. Unfortunately, we don't know how the pattern of virus attachment is in mink, where we've seen likely evidence of mink-to-mink transmission of this H5N1 virus. However, it does belong to the same family of mammals, Mustelidae, as the ferret does. With regards to pinnipeds in harbor seals, not for H5N1 virus, but for other avian influenza viruses, we see unusually that there is good attachment to the trachea as well as to the lung, which could explain why this outbreak could have taken place in Maine. For the South American sea lion, unfortunately, this attachment study has not been done yet, so we don't know there what the pattern of virus attachment is. Again, the South American sea lion does belong to the same group of mammals, pinnipeds, as the harbor seal. Next slide. So in conclusion, we can say that the higher prevalence of high-path AI H5N1 in wild birds is an important factor in explaining the higher frequency of bird-to-mammal transmission, and mainly in carnivores, that we've seen in the last 2 years. Also, there is a moderate to good evidence that there is efficient spread of this virus in some mammal species, the outbreaks that we've seen in harbor seals, in South American sea lions, and in American mink, and that the relevance of these outbreaks for efficient spread among humans depends on the similarity of their respiratory tracts, in particular the pattern of virus attachment of H5N1 virus in these species. And my final slide. So to finish off with the often asked question, why hasn't HiPath AI H5N1 become established in humans? I return to a paper that we wrote a few years ago in which we defined the species barriers for emerging viruses. And in this paper, we showed that these species barriers for a virus to spill over from a donor host to a recipient host can be divided into three categories. A, exposure, B, infection, and C, efficient transmission. And for H5N1 spilling over from poultry to humans, the requirement for exposure has been fulfilled. Namely, there is abundant exposure of people involved with culling poultry worldwide. There are many poultry outbreaks happening at the moment. The requirement for infection also has been fulfilled. Namely, there have been over 800 infections with this HiPath AI virus H5N1 as of 2023. Most recently, unfortunate fatal case in Cambodia. But the requirement for efficient transmission, C, has not yet been fulfilled. There is either no human-to-human spread or only very limited human-to-human spread when there are very intimate contacts, for example, between family members. So to finish off, I'd like to thank, can I have the last slide? The Delta Flu project and the VEO project funded by the Horizon 2020 funds, and also conversations with working groups of the European Food and Safety Authority and those from the AI Impact Working Group. Thank you very much.
Thank you very much, Dr. Quicken. Very insightful presentation looking at the comparative pathology of the different animals, from birds to mammals and also to humans. So some very curious insights as to why avian influenza at the moment still remains not a proficient virus that is transmitted between humans, and thankfully so too as well. So before we start the final presenter, I just want to run the second poll. So if we can run the second poll while I begin the introduction for our final presenter of the day, which is Dr. Richard Webby. So Dr. Richard Webby is a member of the Department of Infectious Diseases at St. Jude Children's Research Hospital and the director of the World Health Organization, WHO Collaborating Center for Studies on the Ecology of Influenza in Animals. He has a basic research program funded by ALSAC, the fundraising arm of St. Jude, and the US National Institutes of Health, NIH, that focuses on influenza diseases at the human-animal interface. This work involves virologic and serologic surveillance activities in animal and human populations to determine the prevalence of influenza viruses present. Further laboratory-based research sets out to understand the mechanisms behind various viral phenotypes. His expertise is in influenza virology, and he has substantial experience in vivo and in vitro models of influenza virus replication, pathogenicity, and transmission. And data collected through the above activity feeds into the WHO GISRS system for risk assessment of circulating influenza viruses, and where appropriate, subsequent pandemic preparedness activities, such as candidate vaccine, virus production, and distribution. Dr. Webby obtained his PhD from the University of Otago in New Zealand, and he has authored and co-authored over 533 peer-reviewed publications with over 25,000 citations. And he's also been awarded several honors and awards. His work is being highlighted in Nature magazine in 2011. As I said, he currently sits as the expert for World Organization for Animal Health, WOAH, and FAO, Food and Agriculture Organization of the United Nations Swine Influenza Expert Group member. And he also, in 2010, received a Special Recognition Award in honor of extraordinary work in H1N1 influenza research by the NIAID, or the National Institute of Allergy and Infectious Diseases, a division of microbiology and infectious diseases. He's also a member of the Influenza Advisory Committee, part of the US National Pork Board. So his research interests lie in influenza virus ecology, influenza vaccination, influenza virus pathogenicity, and the determinants of host susceptibility to influenza. So I bring to you now Dr. Richard Webby.
Thanks so much, Douglas. Is sort of coming at the problem from more of a human perspective, with a little bit of sort of genetics in terms of what we've seen as this virus that we've heard about has made this sort of transition from a virus that for most of its existence has been a problem for Asia, Europe, and parts of Africa, to now where, you know, as we've heard, it's clearly a problem for the Americas as well. And as this virus has made that leap into our continents, it's undergone some genetic changes which have actually changed the biologic property of this virus as well. So we'll summarize that a little bit, and I'll go through this relatively quickly in light of time, and then we'll come back to sort of the human situation at the end. So the story here started actually when Dave Stallknecht sent us over a couple of isolates from early on in the outbreak in the US. As we've just heard from Thijs, that ferrets are sort of the workhorse of the influenza world in terms of trying to model human risk of these viruses, and we set out to just phenotype a couple of these very early licenses, so late 2021, early 2022 viruses in our ferret model, based on experience that we've had over the past couple of decades. Certainly the indication was, at least in my mind, that these viruses probably weren't going to cause a lot of disease in ferret models. So although these are highly pathogenic avian influenza viruses, and if you're a chicken, they're bad news, you know, over the past few years, these 2, 3, 4, 4, particularly B viruses in our hands haven't had much of an impact in mammalian hosts. And the very first virus that we tested was just like that. So we put large amounts of virus into these animals, certainly no symptomology, and none of them met criteria for euthanasia. In contrast, a slightly later virus, this one from bald eagle, actually led to rapid disease progression in these animals, and animals actually had to meet criteria for euthanasia within 7 days. So with clear neurologic involvement. Luckily in this model, we haven't seen we saw transmission of neither of these from one ferret to the next. And to date, we still haven't seen that with any of these H5 viruses. So from a human health perspective, this is good news and potentially bad news. So we've seen some increased pathogenicity here upon movement through, in this case, just North America. But the good news is still no evidence that these viruses have what it takes to transmit amongst most mammalian species, probably including humans. Just a little bit of a refresher here on flu reassortment, in case for those that are maybe not intimately familiar, because this will be important for what I talk about next. What we know that flu does in terms of its ability to evolve, one like any virus, it mutates a lot when it replicates, but because influenza has its genetic elements on individual pieces of RNA, what can happen is when two different viruses infect the same cell, they go about their replication, they mix and match their gene segments, and you can get a slew of progeny virus that comes out of that mixed infection that can have either the two parental genotypes, or they can have sort of a mix of these 8 gene segments derived from, again, either the two infecting strains. And sort of that, from the virologic perspective, the good thing about this is that this can really change the phenotype of these viruses quite rapidly. And again, this is a natural mechanism this virus has. It goes on all the time, and particularly within the wild bird populations that we've heard Dave talk about first up. And this is exactly what we saw when we started to dive into a little bit more detail about the two viruses that were put through our risk assessment model. The first virus, when we looked at the genes, all of the 8 gene segments of that virus, they matched the Eurasian lineages of this virus. So this was essentially the virus that came over from Europe, donated by our European friends. But soon after arriving in North America, it started to reassort with viruses within our wild bird populations. And the second virus that we phenotyped here, this eagle virus, had actually switched out its PB2, PB1, and NP. So these are genes involved in the replication of the virus. But it switched out the Eurasian genes there for viruses from North American wild bird, from viruses from North American wild birds. And when we looked at just a small number of viruses here, we could see a whole range of different combinations of genotypes had emerged soon after this virus came into our continent. And this is not just happening, of course, in the Americas as well. This is something that also goes on within Europe, within Africa, and within Southeast Asia as well. So these viruses are continuing to evolve by mutations, but they're also continuing to sample new genetic material from other hosts of influenza. And particularly in this case, that reassortment event had led to quite a phenotypic change in these viruses, at least in these models, from one that caused little disease to one that caused a lot of disease. And we've done this now with a few more viruses. If we just look at this in terms of ferret and also mice as well, as a second mammalian model. So the closer the viruses come to this end, the more pathogenic they are. The numbers I have here are the number of North American gene segments that they've switched. So again, the early Eurasian ones really didn't cause a lot of disease in ferrets or mice. The virus that had picked up 2 North American genes started to move a little bit down the ability to cause disease, and then through to these viruses here, which are 3 or 4 gene reassortants. And these, at least in our models to date, cause quite severe disease in these animal models. This is a very messy and awful slide. I apologize. I haven't had time to clean it up. But if we look at what's gone on now, this is just within viruses from the Americas. If we look at these different combinations of genes now, and we can see the percentage of viruses representing each of these genotypes. So in this case, the black represents genes that are from those Eurasian viruses that first came over. The red represents genes from viruses from within the Americas. You can see different combinations. There really isn't a dominant genotype to date. So again, these viruses are still continuing to sample some of that genetic diversity. If we look at that here and look in terms of, is there a particular genotype that we should be most worried about? And at this stage, it's pretty hard to tell. And the answer to that is probably right now, they all probably contain the same risk. If we look at particularly the mammalian species we've just heard about, we look at those infections across the sort of spectrum of genetic diversity, we can see that all of these genotypes have infected mammal species as well. So lots going on in terms of what this virus is doing currently as it's moving through, or as it's moved through North America, and of course now down into south of the equator. I will mention that this particular genotype here is the one that's been found certainly in Mexico and most of South America as well. So it contains 4 gene segments from North American viruses. So good news, at least in terms of this virus, this virus still very much maintains avian virus properties. We just heard sort of talk about, this virus still doesn't have the capacity to be expelled from one infected mammal and transmit to the next, or at least in terms of most mammals. These are some of the features that we know this virus is going to probably need to change to make that switch. Avian viruses bind to again, we've heard slightly different receptor than mammalian viruses. All of these viruses maintain the motifs that we know are associated with avian virus-like binding. The HA activation, so the hemagglutinin is this molecule that takes the virus, attaches it to the host cell. This HA has to be activated by pH changes in terms of initiating infections. And again, there's a range, the pH range that avian viruses like versus mammalian is different. So far, at least in the viruses we've tested, the pH of activation all sits within typical avian virus-like. And general genomic characteristics, again, are mostly avian viruses-like. Again, as we've heard, there are a few mutations that have popped up in some of the polymerase genes that can be associated with an increased ability to replicate in mammalian cells. A lot of those have come from viruses sequenced from mammals. There are some of those that circulate in bird populations as well. But from a human health perspective, again, more good news is these viruses really look and smell and taste like avian viruses to date. Although I wouldn't recommend tasting them, I guess. The other good thing from a human health perspective, although humans don't have pre-existing immunity to the H5, or not much pre-existing antibody immunity to H5, the other surface glycoprotein of the virus, the neuraminidase, does share at least the same subtype with the H1N1 viruses, which we know, of course, cause seasonal outbreaks in humans. And we've done some studies can show that the human population does contain a lot of cross-reactive antibodies that will bind to the neuraminidase of the H5N1. And we do know that although neuraminidase antibodies won't block infection, they will block some of severe consequences of infection. They can actually block onward transmission as well. So again, from another risk perspective from humans, no immunity to the H5, but we have substantial immunity to the neuraminidase as well. So should this virus decide one day it's not going to be a chicken virus, it's going to be a human virus, we are going to have some immunity against it. Exactly what that does in the context of a pandemic, I guess, well, hopefully we'll never really know. So despite all that, human health numbers, human infection numbers, we are seeing zoonotic infections are still occurring. This is just WHO data from the middle of 2020 through to actually the middle of last year, but these have continued as well. The H5N1, well, these are all H5s. Most of these have occurred in China with the H5N6. And actually, sorry, I should have already updated this. And actually, I've got the next slide. But we are sort of seeing now some H5N1 infections. And these are the cumulative numbers we've had since sort of 1997 with these H5 viruses. So human infections with these viruses is rare, but we do see them. We do see them every year, well, most every year for the past 20 odd years. What about with these recent viruses? So these the so-called 2344B viruses, which again, this is the form of this virus that is causing most of the infections in bird populations globally, with the exception of some pockets of other sort of H5 activity in Southeast Asia. One case in a person in England, this particular individual had very, very close contact with infected birds, infected almost pet birds. asymptomatic infection. The virus is really only detected because they swabbed this individual pretty intensively. There's been one case in the US in Colorado. This was in an individual who was involved in depopulating an infected flock. Again, was an asymptomatic infection. This person was positive on a PCR swab. When he left the barn the next day, he was negative. So some debate about whether this was a true infection or the PCR just picked up particulate matter in the nose of this individual. 2 H5N1 infections in China in the past 6 months, one of those fatal. All of these individuals have had contact with infected poultry. 2 human cases in Spain. Again, this was similar to the US scenario, involved in some control measures. These 2 were picked up by PCR, asymptomatic. So again, some suggestion that maybe the 2 cases in Spain weren't true infections, but rather just detections of viral RNA in the nose of these individuals. Of course, unfortunately, the more severe case in the young girl in Ecuador that was with an H5N1 virus, and was with a virus that was a reassortant virus. And of course caused severe disease. The 2 recent cases you may have read a little about in Cambodia. This was a father and a daughter. I think the jury is still out a little bit whether this was human-to-human transmission in this event, or whether it was infection from a common source. WHO is still actively investigating this. This particular virus in Cambodia is not the 2344B that has spread widely. It is in the Americas. This is a close cousin, so-called 2321C, which has really been endemic within poultry populations of Cambodia for a few years as well. It doesn't really spread in many other countries apart from a few in Southeast Asia. So clearly these infections are occurring. There's a range of very, very mild to fatal infections. And if these viruses continue to circulate, clearly we're going to continue to see human infections. So what is the WHO and other entities doing about this? Well, one, with a view to pandemic vaccine production. We're not at a point yet where we need to go to full-blown vaccine manufacture. Clearly, we're not seeing evidence that this virus is adapting to the human host yet. But what we do know is that it takes about 6 months to make a matching strain, should that change tomorrow. And the first part of the production system is to make so-called seed viruses. So these are viruses that are manipulated in various ways we don't necessarily need to go into here before they can be sent to manufacturers. So this occurs within WHO laboratories. And this is a sort of a 6-week period. So what the WHO is doing is trying to get a head start on this. And as we see variants of these H5 emerging, we'll actually go through and make that seed virus against some of these emerging strains, just to have on the shelf, so that should they change their mind from being a bird virus to being a human virus, that we've shaved 6 months off of the response. I'm not going to go into this, but if you Google this, every February and every September, the WHO summarizes what's gone on in terms of zoonotic influenza. And when there's an emerging lineage of virus that doesn't have a vaccine seed virus on the shelf, we will make one. This is just a list of all of the H5s. So every line here is a different virus that's been turned into a seed virus sitting on the shelf. Some of these have been turned into vaccine. Some of them we've gone into clinical trial. Most of them, luckily, are just sitting there and have not, luckily, had a need to be used. But this continues every 6 months. So to summarize, within at least the North America, and not just North America, Americas now, multiple reassortments from a limited number of reassortment events have occurred. So this virus come to a completely new area for it. It's now sampling this diversity of genetic material that we have in our continents. Some of them are extremely lethal in experimentally infected mammals. Over the 20 years that we've been studying these viruses or more, some of these viruses are the most lethal that we've seen in these animal models, and some of them particularly neurotrophic. Human infections are still very rare. We need to keep this in context. So you have to try very, very, very hard as a human to be infected with these viruses. And many of the infections that are being picked up are probably not true infections, but more just sort of out of an abundance of caution in terms of close monitoring of people who have been in contact with these animals. But again, rare, but not absent. So the risk is low, but the risk is not completely absent. So we have to be very, very careful. We have to keep following these viruses. We have to keep monitoring populations exposed. Luckily, no evidence of adaptive mutations. And from a sort of a vaccine response standpoint, even though we're seeing quite geographic distribution now of these 2.3.4.4b viruses, antigenically, they haven't moved a lot. So a lot of the vaccine seed viruses that we have on the shelf now still cover most of these viruses well. So I'm going to stop there. Thank a lot of people, and think probably we should move on to questions. Thank you.
Thank you very much, Dr. Webby, for taking us on a journey along the genomics of avian influenza and also influenza these viruses as well. So thanks to all the presenters so far, and now we will turn our attention to the question and answers, as the audience has been so patiently waiting for their burning questions to be answered. I think the first question states, any indication that transmissions have moved up the bird or food chain? And I think, well, yes, from birds to mammals, it seems. Whether it has gone further, maybe the panelists, the presenters can weigh in on this. So the question again is there any indication that transmissions have moved up the food chain?
Within the wild bird community, yes. I mean, pretty obviously. Moving from probably infected ducks to raptors, yes. And also to mammals. Some of the, beyond that, it would be pure speculation. But we don't know the extent of that movement up the food chain, as Dr. Kuiken has said, and it's kind of important to understand that, but that's where we are right now.
Okay, thanks very much. What are the implications of this on bird watching exercises or tours? And then there's also a concern being expressed, or a question posed, are there concerns about human consumption of wild birds that may be infected but not showing any symptoms?
Okay, right now, I think as far as bird watching and that sort of thing, we have not seen any effects of this, except in conditions where there are catastrophic outbreaks going on. And I think there was a situation in South Africa where a sort of a touristy spot was shut down temporarily when they had a bunch of seabird die-offs. But at least in North America, we have not seen anything. And quite honestly, as far as the hunting thing, which gets into the next question, it has not impacted hunting of waterfowl in North America one bit, which is kind of interesting. As far as consumption of waterfowl, looking at our prevalence estimates that we have in wild birds, in ducks, hunter-killed ducks, it can be as high as 20%. And if you consider that the limit of ducks for an individual hunter on a day is 6, it pretty much implies that at least that hunter is carrying an infected bird back with him to the kitchen every time they go hunting, if they're good hunters, that is. That's kind of a scary thing to kind of think about. So we've been kind of addressing this. And again, getting back to what Dr. Webby was saying, it's not no risk, it's low risk. And that is the message we have been trying to pound into hunters, is at least, protect yourself and nothing extravagant, just normal sanitation, but protect yourself. You do not want to be the first case, because that will impact what you like to do. Again, getting back to something Dr. Kuiken said, and about the deep inhalation. And that's what we actually been looking at in terms of hunting, is these little places where that sort of scenario could take place, and they do exist. And one of the things is with a lot of the commercial hunting operations in the big lodges, they have commercial plucking sheds where someone in there will be taking the feathers off of birds and eviscerating birds in a very confined airspace. And we've been trying to get the word out, but I'm not sure we've done any good as far as building a little security around that process. The other thing we've been looking at is sort of the distribution of virus in infected birds. And I know this has been done so many times with poultry at Southeast Poultry. And I know it's been done in Erasmus, but it's a simple thing. It's just basically looking at the tissue burdens, individual tissue burdens, in relation to what a hunter may come actually in contact with. The high risk areas, the brain, the viscera. The lower risk tissues are the skin and the meat. And we're at least trying to get that information out so they can act accordingly. And it's important from a wildlife disease standpoint too, because a lot of when you dress a duck, when a hunter dresses a duck in the field, that viscera, that head may be discarded in the environment. And that's another thing we're trying to communicate with the hunting population, to dispose of those things properly. So the scavengers, the mammals, the vultures, the eagles will not be infected secondarily up the food chain very artificially. So that's pretty much it.
Thank you very much, Professor Stallknecht. And I guess a related question. I just saw you.
Can I add something to that?
Sure.
Kirk? Yeah. So there have been no cases of human infection from wild animals. So I think that's important to note.
Right.
The one exception is a group of women who plucked wild swans in Azerbaijan back in 2006 and actually died from H5N1 virus infection. So it is something that's to reiterate what Dave said, is that something that we have to be careful about. With regards to effect on tourism in the United Kingdom, particularly, several areas where there was tourist traffic to bird islands was stopped because of the outbreak, to actually to protect birds by not disturbing them more. Of concern there was that also in some cases the access of scientists to these bird islands was stopped. So, and this meant that they were not able to record the mortality on these islands. This subsequently changed, so they were given permission to go to these areas, and it's something to be careful about. It is important to try to protect these affected seabird colonies from excess disturbance, but it is also important to have sufficient observations to be able to record what actually has happened.
Dr. Swain, you want to weigh in as well?
Yeah, just to add to what's already been said, is that the avian influenza virus is a very thermolabile virus too. So, you know, if you have, for example, a duck that you're going to cook, you don't know whether it's positive or negative if you take the proper precautions, as Dr. Stallknecht has already said. And when you cook it, the cooking will kill the virus. So the virus, if there's any virus in a carcass, the cooking itself will kill it.
Okay, thank you very much. I see another question in the chat. It's asking about the stability of avian influenza viruses in water, and do we need to treat water if it has been affected? And I think, Dr. Stallknecht, you may want to probably weigh in on this one.
Yeah, this question has sort of haunted me my whole career. These viruses are extremely stable in water. There's no doubt about that. But what you really have to kind of think about here is dose. You know, we've reported in the past that we can isolate it from water, and it's true, we have. But the amount of samples you have to go through to do that is pretty high. It's a low probability event. When you're actually looking at bodies of water, there's a lot of dilution there, especially with the marine environments that I would not be too concerned about. I think treating water would be vast overkill for prevention. With one exception, I would never, ever use surface water that wild birds are using to water my poultry flock. I mean, that's the one exception, because that's when a low risk event becomes a probable event, when you keep doing it and doing it and doing it over and over again. But as far as like human issues associated with environmental contamination, I am unaware of any transmission events that have been documented there. Maybe someone else has a lead on that, but I don't.
Thank you very much. Another question: how can I tell if the bird flu is low or high kind? I guess they mean, is it low path or high path? And what is the treatment for it?
You're talking about in wild birds as far as treatment goes? Well, where we are right now is we're just watching to see what happens. And, I think there has been some talk about vaccine use in highly, highly endangered species and perhaps in zoo. But for the free-ranging populations, it is just not a viable option. I don't know of anything we can do. We have thought about it a while, and we've wrote a manuscript warning people to think about it before it got here from Eurasia. There's no good ideas on really how to control this thing in wild birds, and it may not be possible. And it's really a sad, unfortunate event, but that's basically the story of any wildlife disease. We got limited options. Once prevention breaks down, you pretty much, you can be in trouble. What we really want to do is really understand impact. And that's what we're trying to focus on now is, are there going to be impacts? We don't really know this. So that at least some of these impacts can be mitigated by maybe changing management strategies just a little bit. But that's where we are right now. It's not a good situation to be in.
I guess one of the related questions would be I know in the Caribbean, we have a number of endemic, and some of them are threatened. Some of them may be protected species. And some governments around the region may be, arms in the air as to what should they do with these threatened and protected bird species, now that the threat of bird flu is looming. So that would be some advice there, I think would go a long way for those that are present in the audience as well. I see Dr. Swain and Dr. Quicken. I'm not sure who wants to go first.
I'll just
Go ahead, Dave.
jump in. I'll quickly jump in and say that as far as determining low path or high path, that's usually done by a reference laboratory. But not all countries have a reference laboratory for avian influenza. And the old way to do it was to inoculate chickens with the isolated virus, and you look at mortality rates. Today, though, with the advent of molecular biology and sequencing, we essentially do that by sequencing the hemagglutinin gene and looking at what's called the cleavage site. And if the cleavage site matches a highly pathogenic virus, then it's declared highly pathogenic. That's for H5s and H7s. If it's an H5 or H7 and it's sequenced and has a cleavage site of a low path virus, then it's declared as a low path virus. The other subtypes, the 1 through 4, the 6, and the 8 through 16, there have not been any of those subtypes to be high path, so they're assumed to be low path unless it's demonstrated by inoculation of chickens. And I know that at least in the Caribbean Islands, there's at least one reference lab, that's Dr. Pastor Alfonso's lab in Cuba, that deals with this and sequences, and they can determine that. And there may be other labs in the Caribbean Islands that do that also.
Thank you. Very good information. And I think Dr. Alfonso is actually present in the audience as well. So thanks very much. Dr. Quicken, want to win?
Yeah, I just want to add to Dave Stallknecht's comment that indeed vaccination of free-living wild birds is not an option. However, maybe one thing that can be done is that if there is a high mortality on a seabird colony, it may help to take away the dead birds that are infected to reduce the infection pressure. So in a few cases in the past 2 years, that appears to have helped in the UK, in South Africa, and also in the Netherlands. So that's one thing to consider, but you have to weigh up the pros and cons of doing this.
Thank you very much. This is a question, I think, for you, Dr. Weber. What is the potential for mutation and animal to human transmission?
Yeah, I think we've covered some of that. You know, if we think about what this virus, what we know about what this virus has to do to change, you know, it could be as few as half a dozen amino acids it has to change, which would really change the story here. But it does really seem like these H5 viruses, at least with those receptor binding changes we talked about, which are key to this virus making the real switch, it does seem that the barrier for these viruses to make those changes is pretty high. You know, it doesn't seem, people have, as we've heard, we've sequenced when we see mammalian infections and still no evidence this virus can really make these changes. You know, does that mean it can't do it? No, absolutely not. It just means the barrier to do it is high. So again, the story, yeah, we've said it 1,000 times here. Risk is low, but it's not zero. You know, and that risk could change as the virus continues to evolve.
A related question then, Dr. Webby, is given the propensity for swine influenza to infect humans, what would be the measures for, because I know sometimes even within the Caribbean, we have farms that have both poultry flocks and also simultaneously swine herds as well. So what would be your advice for a situation like that?
Yeah, so we know, yeah, of all of these sort of other mammalian hosts, that swine really are a host that can drive avian influenza viruses to adapt more to be able to infect humans. Yeah, and so at the moment, the pigs do seem to be, and swine seem to be fairly poorly susceptible to these H5, which is, of course, a good thing. But yeah, the practices of perhaps feeding pigs and swine, you know, broken eggs or, you know, dead poultry or stuff like that is clearly not a good idea in this case. So, you know, separating those as much as possible is always a good idea, because, you know, swine really are a risk animal, a risk population for humans with influenza for sure.
Great, thanks very much. I think the next question is looking at the SPS measures for the importation and export of meat that could be possibly contaminated or infected with avian influenza. The question is, has there been increased controls over exports and imports on meat? And I'm wondering if this is from North America. I believe the question is. Perhaps Dr. Swain, you can.
Yes. So every country that exports poultry, they would have an agreement with the importing country that would indicate what their surveillance program is for example, avian influenza, but other diseases such as Newcastle disease, and that would be the basis of allowance of importation of products. Now, some countries allow regionalization. So, for example, if we had a case of high path AI in Maine, it would not impact products that are in another state like Georgia or something like that. So, and also it depends on the country. So those agreements decide what surveillance data you have and how small you can make that zone where the infected product is. When there is an infection in poultry on a poultry farm, there's also at the same time as putting down that affected flock, there is also surveillance done in a zone that farm is in. Usually it's a 3-kilometer zone, and also outside that would be another surveillance zone. You would be testing all the poultry to demonstrate they're either free or they're positive, and that also adds the assurance that what you would receive is free. And I think when I mentioned in my presentation is that in the U.S. for us, it doesn't matter whether you had a vaccinated flock or non-vaccinated flock, you would still that product would have to be free from influenza to be put into the food process, the food chain. So we would not have a vaccinated flock that was positive for influenza and then put in the food chain. It would basically not be put in the food chain.
Hope that helps. There's a question from Chris. She's from Aruba. She's the chief veterinary officer there. She said, if I'm not mistaken, there are reports of brown pelican infected with HPAI very close to Aruba. What are the chances of avian influenza, the hypopathogenic avian influenza, coming to Aruba with these birds? And I guess
Yeah, we've had quite a few brown pelicans in the US and I believe pelicans in Peru and other places. Be quite honest, I would think it'd be pretty good, but I do not know. I would have to look at the behavior and the migratory patterns of that species specific. And off the top of my head, I don't think I know that. But there are plenty of seabirds that migrate. And I think the probability that an affected bird would make it anywhere in the Caribbean is probably pretty good.
okay, thanks very much on that. There's another question here about the capacity. Is there capacity in the Caribbean to vaccinate susceptible bird populations? I'm assuming this is poultry, particularly concerning wild chicken populations that run wild in neighborhoods and may have close contact with other domesticated animals in non-farm environments. So I guess the question is regarding the capacity, because if for instance, vaccination is one of these strategies that we can potentially employ, the question is that does that capacity really lie within the Caribbean to vaccinate these large commercial flocks within the Caribbean?
I'll take a crack at that. So, poultry production around the world for human food consumption in agricultural processes, there is already in place for all the commercial operations to vaccinate for endemic diseases. So whether they're given by spray, water, or injection, the process of vaccination is already there. That's not the issue. The issue becomes, is the vaccine available and is it approved by the country's government for use? And that would be the part that I think the Caribbean islands would have to, on an individual basis, decide if you want to vaccinate or not vaccinate. From my limited knowledge of the Caribbean islands, I think the Dominican Republic has had a vaccination program for H5N2 low-path AI for some layer flocks within that country. So there is experience with avian influenza vaccination in the Caribbean islands, and it will be an individual country decision. The other challenge is that there is in place in any place where you have a commercial poultry sector, vaccination is used for endemic disease. So you have that process. The issue becomes in village poultry, which may or may not be vaccinated. Is there capacity there and could it be done in village poultry? And that is something that I think most people don't know. In most countries that are vaccinating for high-path AI, generally smallholder poultry, village poultry, backyard poultry are not included in the vaccination programs, or usually they're an exception to the vaccination, but usually it's in the commercial poultry that are doing the vaccination.
Okay. I guess one other question could be, if a flock is vaccinated, does that render the flock with sterile immunity, meaning that should they be exposed to any strains of H5N1, would they then be completely protected, or would they also have some protection, but also there might be a reduced risk that some of those chickens could become infected?
Yeah, so vaccination by itself reduces risk. I think I know I've read quite a few papers where people talk about sterilizing immunity, and sterilizing immunity is only something you have in the laboratory. You don't have it in the field. You just reduce the ability of the poultry to become infected. And if they do become infected, you reduce the ability for the virus to grow. Some of those statements about sterilizing immunity are based upon a single animal experiment with a certain challenge dose and then the sensitivity of the assays that try to recover the virus or detect the virus have a different level of sensitivity. So when a person makes the statement of it's sterilizing immunity, they're that's a loaded question because it's based upon a set of data that is very much limited in what that data says and how it's done.
Okay, thank you. And the other question was, are there vaccinations available for this strain of H5N1 currently?
Yeah, around the world, there are various biologic companies that do make vaccines that closely match, either identical or very close to that, but not all companies do. And so part of the issue is if you decide to do a vaccination program, you will have to contact the company that would be licensed for a vaccine in your country, and then they have to make that vaccine. They don't always have it on the shelf. For example, in South America, a couple of countries, Colombia, Peru, and Ecuador, are looking at that vaccination, and Bolivia, looking at vaccination, and they can purchase it, but it may be 2 to 3 months out before they get that vaccine that has to be manufactured for their use.
Thanks very much. And I think we'll try to wrap up the entire proceedings by 3:30. I think we've been going for quite a while. I know we have a number of burning questions, but I do believe these gentlemen have some other stuff to attend to. So we will try to make it quickly and see if we can make some rounds. So there's a question here in terms of perhaps using lower densities, particularly for the production of whether this be poultry or even in the case of mink, the mink farms, because when you have large farms with high density of one particular animal, then this seems to be a vulnerability, if you would. Is there any benefit for keeping birds at lower densities as part of a multi-pronged solution?
So in poultry production, of course, we in the United States have had 58% of our premises that are positive on the poultry side are backyard flocks. So just to make sure you understand is that chickens and turkeys are very susceptible to this virus, and it doesn't matter what density you have in that. Now, you may decrease your odds of getting exposure and transmitting between farms if the farms are closer together. But the vast majority, at least the early studies done in looking at transmission, have been transmission to poultry farms have been from environmental sources and not onward transmission from affected farms. So the density of the poultry has not at this point impacted the transmission in our setting in the United States. In some countries where there is transmission from farm to farm, they have looked at reducing the number of farms. We're not talking about reducing the number of poultry. They've looked at reducing the number of farms in a geographic area and make it more of a low density. And that could reduce the transmission, but you'd also have to find other places to put those poultry to meet that lower farm density, not animal density.
Okay, thanks very much. There's a question here regarding, given the practice of persons going to the beach and among the birds, particularly the fishers, is there any concern for human infection from contaminated water, et cetera, at the beach?
So let's hit the beach first and the fishers second. We've actually done some environmental sampling in Delaware Bay associated with shorebirds. And this is a really super high contaminated areas. These are right under the net, thousands of birds with a high infection rate. And you can recover it from the sand. But again, it's just really similar to what I said before, that you really got to work at it to recover it. I mean, it's there, but it's probably at a very, very, very low concentration. And this is detectable in 24 hours. I don't know if it would be detectable after that. We never really followed up on that. So it's possible to get viable virus from sand, but this is a situation that just in a small area that's just covered up with known infected birds. As far as the fishermen go, I spent one summer working on a shrimp trawler off the Georgia coast. So I know what they're talking about as far as a lot of birds coming up with when you pull the nets in, et cetera, et cetera. But I personally, and I'll just give you my personal, I personally would not feel threatened by that. as far as a high-risk situation. A lot of birds, but the contact that Dr. Kuiken talked about, the actual contact that needs to take place probably is a rare event at best.
Thanks very much. What are the consequences that this highly pathogenic avian influenza virus can have on not only on animals as well as businesses as it relates to food security? So I guess this is more in terms of what is the threat that bird flu, the highly pathogenic form of it, poses to food security, and when and where was the latest case of this virus detected, if you can recall?
Yeah, I answered that in the chat, but I'll just summarize. So there are cases every day being reported for this virus around the world, US, Canada, and in multiple Latin American countries. So the last case that was reported most recently is going to be today, probably. It's going to be in multiple countries. I guess probably the most recent cases that have made a headline have been the mammal cases, and there's been some wild bird cases reported in Uruguay and Argentina, which is on the eastern side of South America, which is a new barrier that's been crossed historically. I guess that Davidstalnick can talk, but historically there has been a barrier of the Andes Mountains and the Amazon basin that has sort of stratified low-path viruses. So there is some genetic differences between those two sides of South America, and that's been breached already also. But those are reported just about daily. There are cases reported around the world. I guess the second part was food security. So I think I made a comment that for us in the United States, the numbers of egg-laying chickens that have become affected in those farms to be populated has reduced the supply of grade A shell eggs, and eggs are consumed heavily in the United States as they are throughout all of the Americas. We have a shortage of eggs, and with the supply and demand, the price has changed dramatically. Pre-outbreak, they're running a little over $1 a dozen, $1.50 a dozen. The other day, my wife went and bought some eggs, and it was $5 for a dozen of eggs. So that's an example of food security, in that if you are a low-income person and you're at the grocery store and you have a low amount of money, you're probably not going to buy the egg as your protein source because the cost is so high, and it could put you at that person at a nutritional issue of not having enough protein long term. Eggs are great thing about eggs is that they can be non-refrigerated for a period of time, so they're easy to in very rural areas without reliable electricity. They can be maintained as a food source of high protein quality protein source. And so if you lose that source because of cost, it can have nutritional impact.
Thanks very much. I think this is a question from Dr. Alfonso. He's asking about the mutations in the viruses found in the minks in Spain. I'm wondering if the mutations found there occurred prior to the outbreak or due to the massive multiplication in these animals. And I think this is probably for Dr. Kuiken.
Yeah, so those viruses were compared to the most closely related viruses in wild birds, and those viruses in wild birds did not have those mutations. So the most likely explanation is that the mutations occurred in the mink.
Okay, thanks very much. Dr. George is asking about a coordinated surveillance program for H5N1 in the Caribbean and where it's centered. To my knowledge, I know that there's CARVET, which is the regional animal health surveillance unit. So you may want to check that out, Dr. George. Is there anything you know about similar viruses in reptiles? I don't think so, but no. Yeah, I don't think so. There's a question related to mRNA vaccines and whether these are currently being trialed from Professor Ura.
I think maybe a couple from a human health, but talking to human health perspective, yeah, certainly there are, sort of before COVID came along, a lot of the mRNA companies were developing flu vaccines. Certainly there was one that was hoping, I think, to get approved this year. Some of their original trial data was a little bit disappointing for influenza B, but these companies are also developing H5 monovalent mRNA vaccines as well. As far as I know, none of them are yet been in the clinic, but they're undergoing preclinical testing. I'm not sure about from the chicken world, David.
Yeah, and there's one biologic company that has an mRNA, a self-replicating mRNA vaccine that they've been trialing in European Union. It's not approved in the US, and I'm not sure where their licensing approval process is in Europe, but there is one that I'm aware of.
Thanks very much. And there's a question here about the probability of transmission of avian influenza from wild birds to pigs and from wild birds to commercial poultry could be quantified. What would be the specific value? This is from birds, wild birds to pigs, and wild birds to commercial poultry. Anyone wants to?
So I can answer for that maybe for the Netherlands. It's difficult to actually give a absolute risk for an absolute number to the risk, but you can compare the risks between different, for example, geographical regions, and for example, between different species of poultry that are being kept. And in a recent report in the Netherlands, it was found that the risk for an infection on a poultry farm was much higher, significantly higher in areas where there were many water birds compared to areas where there were less water birds, which makes sense, but has now also been shown by this such a risk assessment. Similarly, the risk for turkey farms and domestic duck farms was several times higher than for comparative layer hen farms. So there also, you can see that the difference in species that you're keeping makes a difference for the risk of incursion of high-path AI into the farm. In this case, most likely coming from infected wild birds or their environment.
Thank you very much. I want to make sure I haven't left out anyone's questions. It's a vaccine and trade is a major concern. Any thoughts on this? And I guess this is surrounding the current ongoing debate of whether to vaccinate flocks, how that will influence potential export trade to external markets.
Yeah, that is the main reason why some countries are not vaccinated for high-path AI, is the concerns about trade. And it actually is summarized down to surveillance and the guidelines that are written about AI control say that vaccination should not interfere with trade if there's adequate surveillance. And I think right now we're in that process of trying to define what adequate surveillance is, and then countries discussing it between importing and exporting countries of what surveillance is required. I think the concern is that if you import product from vaccinated poultry, could you have high-path AI in there? And I think I made the comment earlier that well-vaccinated flocks will not become infected, and the risk of them becoming infected is greatly reduced from a non-vaccinated flock.
Okay. Thanks very much.
Yeah, just to add to what Dave said about surveillance, the European Union last week published regulations harmonizing the required surveillance for high-path AI in the poultry sector in European Union countries. And it consists of having monthly checks by an official veterinarian of the poultry holding, having weekly checks of any poultry found dead in the poultry holding for AI, and if birds are allowed outdoors, also to have monthly checks of the poultry, either serologically or virologically, for presence of avian influenza. So that's the level of surveillance that is now being required in countries of the European Union.
Interesting. Thanks very much, because I know that's a really burning question and one that has taken the global attention, particularly on avian influenza, as we struggle to find a way forward in terms of how do we deal with this emergent biosecurity threat. I think on behalf of the Center for Biosecurity Studies and also the University of West Indies and the Faculty of Social Sciences, I really want to profusely thank all four of you gentlemen for taking time out of your busy schedules. I know that at this point in time, this is a major concern, and you have been working extremely hard for decades before, and you'll continue to do this human service to the entire globe. And so I really want to say a heartfelt thank you for making the time today to answer the questions and also to provide some really important information for those who would have joined and those who will also have access to this recording, not only within the Caribbean, but by extension also perhaps Latin America, both Central and South America as well. So I also want to give special thanks to the team at CITS who have arranged this webinar and have fine-tuned all of the audiovisual quirks that we may have had and supported us in bringing this wonderful webinar event to you, and also to the team at Marcom, arm of the University, Marketing and Communications, for distributing and also having such a wonderful, wide, broad audience that we had today. I think we had over 150 attendees, which is really unprecedented. And so we're really thankful. It just shows you the importance of this particular theme and this important issue. And we just have one or two other housekeeping matters. We do have some upcoming webinar events. As you would know, the 3rd of March marks the World Wildlife Day celebration. And while we don't have an event on the 3rd of March, we do have one on the 6th. And this particular webinar event will be brought to you by Dr. Mark Gibson. He is the director of Nature Nurture. This is a small NGO in Trinidad and Tobago that is looking at and examining the pet and aquaria trade within Trinidad and Tobago, and also the illegal wildlife trade. And again, this poses a really salient threat to Trinidad and Tobago because of this activity. And so he will be delivering some of the research findings that he had from his thesis. And I think it will be a very exciting event as well. And at the end of the month, we also have a continuation of our climate and sustainable development webinar series. So you can join us for that event as well. And we will also share with you the particular details on those two events. And once again, I want to thank all of you who joined this webinar. And I really want to sincerely thank everyone for participating, even with your answers. Your questions, sorry. So on this note, on behalf of Janice and myself, I want to bid you all a good afternoon, and I hope you guys take care and stay safe. Thank you very much.
Sure.
Bye-bye.
Goodbye.
Bye-bye.